Hello, and what I'd like to do today is a really quick video on Unit 3. This is going to be a study of the objectives, the three sets of objectives for the Unit 3 cell test. And you can see that the three: cell theory and microscopes, organelles, and cell types and cell specialization. We'll do speciation soon. Okay, 3A, identify the parts of the microscope and focus a slide. Can state and explain the parts of the cell theory and can describe key discoveries that led to the development of the cell theory. Organelles, you can identify cell structure and functions like nucleus, ribosomes, endoplasmic reticulum, Golgi bodies, mitochondria, chloroplasts, lysosomes, vacuoles, cell membranes, cytoplasm, cytoskeleton, cell wall, and centriole. And cell types, compare and contrast prokaryotic with eukaryotic, compare and contrast plant and animal cells. Can you sequence and define the hierarchy of organization of cells, starting with atoms and then molecules, cells, tissues, organs, organ systems, and organism? And can you describe the importance of cell specialization and multi-cell organisms? So the first thing is we're going to go to a picture of a microscope and talking about the ocular, which starts the magnification process. And telescopes and microscopes both carry light to see images. Your light source is down there. Unless you have a dimmer switch on this one, you've got a diaphragm which is going to control the amount of light that actually goes through your slide, or through the hole in the stage to your slide, so the objective can see it. You've got clips that actually hold it. Coarse, focus, raises and lowers this slide or this stage with great motion, and then fine adjustment for smaller motions. You can actually click this and go to a virtual microscope. But anyway, the second thing that does the magnification of the objectives here on this revolving nose piece, and you've got low power, medium power, and high power, and this is probably a 4, probably a 10, probably 100, and this is probably a 10x, and you multiply the two numbers together. So if you were looking under high, you'd have 100 times 10, so it's 1,000 times. You do the 10 times 10 is 100. Do the 4 times 10, you get a 40 magnification. You always grab the arm and hold under the base when you move these things around. Cell theory. Historically, all living organisms are composed of cells. They can be either multicellular or unicellular, one cell or multi-cells. The cell can be the basic unit of all life. If you're a unicellular creature, that one cell does everything you need to survive. We did a dog racer earlier, which I think you can see in my Unit 1 review. And DNA is actually going through all those things, DNA all the way through reproduction. That individual cell's got to be able to do all of it. And then for our case, we have individual cells that do specialized purposes, and we'll talk about that in the last slide. Cells are the basic unit of life, and all cells arise from pre-existing cells. Now we have a more modern cell theory, and this talks more about things that we've discovered since the 16th century, the 1500s and back. But basically energy flow occurs within the cells. This is the smallest unit of living things, and the energy that we need from either sunlight or food that we eat starts at this process and ends at in that process. Heredity and information. The DNA is passed from one cell to another, and all cells are the basic. All cells have the same basic chemical composition. So if you're a unicellular creature, you've got to be able to do the same thing as a multicellular creature. Maybe not as specialized, maybe not as well, but you've got to be able to do everything a multicellular creature can do just by using your organelles. So chemical composition, whether you do cellular respiration, which all cells do, it doesn't matter whether you're a unicellular creature or us, 60 to 100 trillion cells, we do cellular respiration the same way. Okay, the cell theory was actually made by help of quite a few scientists. We didn't talk about Jensen, but back in the early 1600s, he comes up with the whole idea of a compound microscope. We're using two lenses. Didn't look anything like this. That actually was down here in 1886, where we got the modern compound microscope, even though that's not quite as modern as what this one was. But he makes the first microscope. 1663, Robert Hooke, monk, comes up with the word cell. He actually discovers cells in a dried up cork, which I'm sure they use to bottle their favorite beverages. About 10 years later, Leeuwenhoek comes in and he actually discovers multicellular creatures and looks at living cells. Then we come over here with Schleiden. Schleiden realizes that all plants are made of cells. A year later, Schwann says that all animals are made of cells. A few years later, Virchow actually says that all cells come from other cells. And then we have the compound microscope, and then we have the electron microscope, and then we actually have down here, and it says, will you be the next famous cell scientist somewhere in the future? Then we talked about cells, talking about the difference between plant cells and animal cells. And you can see we have a cell wall, a cell membrane, a vacuole. Cell wall actually gives support and stability. Cell membranes can control things that go in and out of that cell. Vacuole, big storage of water. Plants wilt. It's because this thing actually shrinks. The water gets utilized inside the cell, and then when you water it, these things get puffed back up, and it is the support for the plants. The nucleus, which is going to control everything that goes in there, as well as having the DNA. Nucleolus, which is going to make ribosomes, and ribosomes down here are going to actually put proteins together, and cells are made out of proteins, and organisms are made out of proteins, so that process is actually fairly important. Then we have nuclear membrane, which controls things in and out of the nucleus. We have chloroplast, which Lynn Margulis actually says was a prokaryotic cell that went through a process called endocytosis into the cell and has stayed in there. It does photosynthesis really well, and supposedly the cell gives this thing everything it needs. And evidence she used is actually it talks about mitochondria and chloroplast as being a double membraned cell. It also has its own DNA, and they actually reproduce on their own by pinching, which is the way prokaryotic cells do it. They don't actually have to separate their chromosomes through mitosis. They actually just kind of grow, pinch off at the center, and make two brand new cells. And then mitochondria, the powerhouse. Golgi bodies, which package and transport, or they actually bring in packages and cut them up for transport either in the cell or for out of the cell. Ribosomes, like I said, make proteins. Smooth ER moves things around primarily from the nucleus to the cell. And then we have rough ER, which actually have ribosomes on it. So we have free-floating ribosomes, which you can see in the cytoplasm, and you can see the ones stuck to the endoplasmic reticulum. They can actually work with proteins and put them inside this endoplasmic reticulum to transport them around. Then we have centrosomes. Then we have amnioplasts, which we didn't talk about, neither one of those. And then we have the cytoplasm or cytosol. Then we get to the animal cell, where this is more rigid because of the cell wall. Animal cells don't have the cell wall. All they have is the cell membrane on the outside, so they can take a more amorphous shape. Usually they're rounder or more oblong or more circular than what plant cells are. And here we have a penocytotic implant where something is actually coming inside of the cell. It's too large to come into the cell membrane, so what happens is we get this fold. The material moves in, it pinches off, and actually is surrounded by a membrane, moves in sort of into the cell. Lysosome, which is going to release digestive enzymes if something invades the cell or something is broken in the cell. This will digest it, break it back up into the four organic compounds, which I'll talk about in a second. We have smooth ER, we have rough ER, we have a cytoskeleton, which is also in the plant cell. Cytoskeleton, where is this? Do they tell us anything about cytoskeleton? This one doesn't say anything about the cytoskeleton. It comes in two forms, microtubules, and it does talk about microtubules, and doesn't show the other part. But anyway, we got ribosomes free floating and stuck to the endoplasmic reticulum, making the rough endoplasmic reticulum. We have cytoplasm or cytosol. Animal cells have centrioles. We have a nucleus, we have nucleolus to do all that, we have a Golgi apparatus, we have mitochondria for power. So looking at the two cells, you can see there's a lot of similarities. Things that are different is plant cell has, this is actually gets you to cells alive, which is a project my kids did. Plants have chloroplasts. They also have a cell wall. Animals have Lysosomes, the digestive enzymes. Plants have to be able to do it too, but they don't seem to have any membrane-bound organelle that does that. And then animals also have centrioles to help us with mitosis. These will actually create the spindle fibers, which will grab onto the condensed chromatin into chromosomes. Plants do very similar things, but they don't seem to have the centriole to do that. So those are the two things found in animal cells, not in plants. These are the two things found in plant cells, not in animals. Then we talked about the difference between prokaryotic and eukaryotic. The first thing is that prokaryotic cells are very small. Second thing is that their DNA is more circular and not really inside of an enclosed nucleus. So they've got more of a nucleoid region. They both have ribosomes. They both have cytoplasm. They both have, I think that's it. So we have very large eukaryotic cells. Eukaryotic meaning they have a nucleus. Prokaryotic means they're before the nucleus. But remember, they still have to have DNA. And then the second to last thing where we talk about atoms. Atoms are made out of chinops. Carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Put those together for organic compounds, which we call proteins, lipids, nucleic acids, and carbohydrates. We put those together to make cells, so they're actually made out of the same four things that these molecules are made out of, which are made out of those 6 atoms. Then we go to tissues, which are made out of cells, same things. Then we have tissues coming together to make organ systems. In case this would be like the stomach lining, actually has cilia on the outside to move food around. This is the stomach, and we put the stomach in with other organs to make organ systems like the digestive tract, starting with bringing the food down in the esophagus, into the stomach, into the small intestine, into the large intestine, and out the anus. That's the digestive system. And then we can put digestive systems, respiratory systems, circulatory, skeletal, skin, put those all together to make the organism. So we're getting more organized coming this direction, and each level is made out of something that's in the past level. All of them made out of these 4 organic molecules: proteins, lipids, nucleic acids, and carbohydrates. All those made out of chinops: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And the last thing is structure and function. If you're a multicellular creature or an individual unicellular creature, your shape is basically defined by what you're going to do. This cell has actually got a flagella. Flagella is for whipping back and forth for motion. So this cell actually has a nice hydrodynamic shape, so it can move easily through the water. This is a nerve cell. Nerve cells work by connecting to other nerve cells and sending messages, and you can see it looks like it can attach to lots of different nerve cells, going to arms and body and legs and left side and right side, and then moving up toward the brain. So function, structure, and function. They have the structure is defined by the function of the cell. Okay, that's it. Thank you much for stopping by. Hope that helps.